Hi, everyone. Thanks for joining ahead of time. This is Meredith. I just wanted to say that we'll get started with Gagana's presentation in a few minutes, probably a minute or two after the hour. You will see in your GoToWebinar panel that you have a questions box. So if you have any questions along the way, feel free to put your question in there. If it's like problems with the video or an audio issue or something like that, we'll have Rebecca here to help you with those issues. If it's something about what Gagana is presenting or about filament tracer and working with neurons in general within Imaris, we've got Dieter here from the European support team to help answer questions as we go along. And then Gagana will answer a few questions at the end with me for everyone to hear. So go ahead and get those questions in there, and we will be back in just a few more minutes. Thanks for joining us. Hi, everyone. Thanks so much for joining us. I just wanted to drop in and say hi and let you know that we will be getting started in a few more minutes, probably a minute or two after the hour. Thanks. Hi, everyone. This is Meredith, and I am here just to introduce this next presentation in our Imaris Homeschool webinar series. So today we're going to have Gagana giving an introduction to Filament Tracer. And so she's going to go through a lot of information here. It's a great presentation. I think you'll learn a ton. If you have any questions as she goes along, feel free to use the question box. We've got Dieter here from the European support team to answer some questions or to answer those questions. And then we'll also go over a few at the end of the presentation. So thank you again for joining us and taking the time to learn more about Imaris. If you're interested in anything else about Imaris, go ahead and put that into the question box as well, and we'll make sure you get to the right person to help you out. So I think without further ado, we'll get started, and yeah, thanks.
Good morning, good afternoon, good evening, everyone. Thank you for tuning in today to our Imaris Homeschool webinar series. My name is Gergana, and I'm the Imaris Regional Sales Engineer for the Nordics and North and Northwest Germany. This and next week's webinars are devoted to our Imaris Filament Tracer module, where today I will introduce you to the core functionalities and applications, and next week my colleague will dive deeper into the more complicated cases. And also, hopefully by the end of this webinar, I will be able to show you and convince you that the filament tracer can be used in many different fields of research and not only neuroscience. So when should we use filament tracer as opposed to other models available in Imaris? Suppose we have a cell that has some projections, just like this beautiful endothelial cell. So the first question would be, what type of analysis are we interested in? Are we looking to measure the volume or the surface area, its shape in terms of ellipticity or sphericity? Are we interested in its intensity, or are we looking to do some native distance measurements between the cell and these nice blue objects that we can see associated with it? In case we answered yes to these questions, then surfaces would be the go-to model. However, if we're rather more interested in the length of those projections, the diameter, the straightness, if we're looking to do some branch hierarchy analysis, or if we're looking to measure the angles of branching, the number of the branching points or ending points, if we're having a neuron and we're also looking to classify the spines and study their density, then filament tracer would be the model of choice. What structures are best modeled using the filament tracer? The most common type of structures that springs into mind when thinking about filaments are neurons, and more specifically their neuronal arborization. And as the Latin root of the name suggests, we are talking about tree-like structures or cells, and these are not only neurons, including their spines, but these can be any type of cell that has long processes, such as endothelial cells, like the one that I showed in the previous slide, astrocytes, microglia, and so on. And the second type of structures that we can model are network-forming ones, such as vascular networks, mycorrhizals, mitochondrial networks, and so on. And even though both types of structures have the common feature that they can be defined as filamentous, they do differ quite a bit in their underlying architecture. So what I mean by that is that, for instance, in our tree-like structures, here we have a definable start position, okay, in contrast to our network structures where there really isn't one. In order to be able to segment those different types of structures, there are two different types of automated algorithms available in Imaris, and we can choose depending on the type of model that we're looking to generate. The first algorithm that is available to us in Imaris is the autopath algorithm. It produces tree-like filaments with definable starting position. As I mentioned, generally that would be the soma. And the way that the path is determined is by connecting the large starting point and a smaller seed points. And the second automated algorithm that is available in Imaris is the threshold algorithm, where it produces filament with loops. Again, so when there is no definable start position, here as is in our network structures example. And the idea there is that we would create a mask using a certain intensity threshold, which is then thinned down until we're left with a single center line or a skeleton. And today I will go over these two algorithms, and I will show you exactly what that means and how that looks. So before I move on to Imaris, I would like to just take a minute to briefly mention some examples and applications where filament tracer has been used quite extensively. Of course, firstly, in the field of neuroscience, in the study of neuronal connectivity, where we would like to study the branching patterns of neurons, such as their polarization, the dendritic complexity and length. Further on in developmental neuroscience, as well as if we're studying learning and memory formation or Alzheimer's disease specifically, we would be really interested in investigating the dendritic maturation and synaptic functions. And the best indicators there are spine morphology and density. Furthermore, for those who are in the fields of neurodegeneration, such as Parkinson's disease and brain injuries and brain traumas, we would be quite interested in detecting the inflammatory responses in the brain. And that is best done by performing morphological analysis of microglia, and we can then base that analysis again on filament length and complexity. Again, quite important topic in the field of neurodegeneration, but also aging, that would be the study of mitochondrial health. And statistical parameters of interest would be the average length of the mitochondria, as well as the branching points. And finally, I will just briefly mention some applications in cancer and stroke research, where scientists are generally mostly interested in the quantification of the vascular architectural abnormalities after injury, and they can study that using different statistical parameters, such as, again, diameter, volume, and the capillary density. Of course, this is not an exhaustive list of applications, as this model can be applied to a wide spectrum of scientific questions. So that being said, I would like to conclude my PowerPoint presentation. And what I would like to do right now is to share my screen, where I have already opened Imaris, where we can start looking at the first example. Okay, so I have switched here the screen so that we can see the Imaris interface. And what we have visualized here is this hippocampal pyramidal cell that's in organotypic cell culture. And in this specific example, I'm interested in studying the general dimensions of this neuron, such as its filament diameter, the length. I would like to also perform some branch analysis, such as branch hierarchy. And finally, I would like to detect and classify some spines, since here the resolution is good enough for us to be able to really detect these spines. And in order to do that, I will click here on this Add New Filaments button. And immediately you can see here in the lower left-hand side that this command starts the filament tracer creation wizard in Imaris. And already in the first step, we can choose which algorithm we would like to use. Again, there are two algorithms, so the autopath and the threshold. And if by any chance you don't remember the characteristics of those two different algorithms, you can click here in the drop-down menu, where you can find this nice description of both algorithms. So in this case, we are working with a tree-like filament. As we can see, we have a starting point, or we will detect a starting point, and we have our branching dendrite, so we're definitely working with a tree-like filament. That being said, we also would like to calculate the diameter of the filaments from information from the image, basically the image data. And I will definitely also keep this option here. Now I will click next. And on this next step, what we need to do is we need to select the source channel. So in this case, there is only one channel, so this would be channel 1. If there were additional channels, I could then click here on the drop-down menu and I could select for the channel that I would like to use. And the next point here is that we need to also put in some values for our starting point diameter and our seed point diameters. So if you already know the values or the average value for the cell soma in your samples. You can already go ahead and write that number in this field. However, for those of us, let's say, who aren't sure, what we can do is click here in the slice mode. And what I can do is, using the left mouse buttons, I can click here, and if I click again with the mouse left button, I will then also get the second measurement point, and I can again move this measurement point to refine this distance calculation. And for instance, in this case, the value between these 2 points that I have just placed is 8.4. So I would go back with that in mind. I would go back here to the 3D view and just put in that number. So let's say 8.4. Similarly, I can also use the same idea here to calculate the seed point diameter, so which would be again defined by the thinnest diameter or the dendrite ending. Okay, so I think this value here, it's quite good. So I will keep this for this example, and then I would click next. And what we have right now is a lot of seed points and a single starting point. And just like the other wizards in Imaris, here we have the option to increase or decrease the thresholds for both seed points and starting points. And when it comes to the starting point, here already the automatic setting was already correct, so I will just keep that under the automatic mode. However, for the seed point threshold, this is a bit more delicate manner, so to speak, because here what we need to do is we want to make sure that we have enough seed points here, that we also have detected the ending points of the filaments. At the same time, we don't have too many seed points here that have been detected at the local intensity maximas originating from the spines, as you can see these ones here. And I also don't want too many seed points, because the more seed points I have, the longer the calculation would be, and the more errors can be present in our final segmentation. So it's really a matter of finding this sweet spot, or rather this Goldilocks situation, where again I don't have too many points, but I also don't have too few. And what I can also do here is that if I want to add or delete a seed point, what I can do is press down the shift button and pick a spot. Then with the left mouse button, I can delete that seed point like so. I can also add a seed point by clicking. Again, what you saw here was that I pressed the mouse left button, but it was not directly on the seed point, so it actually created a new seed point instead of deleting this one. But yeah, if I want to delete a seed point, I would click on the left mouse button, and also if I would like to add one, I would also click on the mouse left button. Now this also works for the starting point, and in this case, again, I'm just pressing on the shift and in this case right mouse button, and then shift and right mouse button to create a new starting point. Okay, here we also have the option to remove the seed points that are around the starting point, and the reason for that is that in some biological samples there are simply some protrusions or some signal that we're not really interested, that just emanates from the cell soma. And this is why we have this option to remove all the seed points that in this case are with diameter of the sphere region. So basically at 17 microns distance from the sphere, all seed points will be deleted. So this is a quite useful option. I would say I keep it selected, even though in this case we don't really necessarily have these protrusions, the set protrusions. And the other option here that we have is we can remove disconnected segments. And this is useful in cases where we have, let's say, some projections that are coming from neurons that are not the neuron that we are interested in analyzing. And there is again some projection that's just in the field of view, but we don't really want to detect or connect any of our seed points with the seed points that are coming from this signal. So by checking this option, what we can do is we command Imaris to isolate or to just not connect that type of signals to the neuron which we're trying to analyze, and that would be based on distance threshold. And in this case, again, I don't really have such signals, so I don't need to have this option ticked. So I will then proceed on to the next step. And at that point, this is when the autopath algorithm kind of is being performed, where the tracing algorithm connects the starting point with the end point via the shortest path. And here we go. So this is the final result, or this is the result at least when it comes to the path. And now the next step would be to measure the dendrite diameter, because this is what we have ticked. If we didn't want to measure the dendrite diameter and we're strictly only interested in, let's say, length of those filaments, then that would have been the end of the wizard. But in this case, we also want to calculate the distance. So what I will do is I will just quickly reset the image here. And there are two different algorithms here that will help us to calculate the diameter. One of them is the shortest distance from distance map, and the second one is the approximate circle of cross-section area. So what these mean, here I will show you a very nice schematic. So what I'm doing right now is I am right-clicking with my mouse on the algorithm, and I can now choose the show help option. And what I get is these very nice schematics here that just to get a better representation on how the diameter is calculated according to the different algorithms. So very briefly, this gray or this blue crossed area, this is essentially the mask that we have created here in gray, right? And the yellow disk here, so that would be the filament center axis that we just calculated in the previous step. So in one case, the algorithm would work by trying to find the shortest distance from this path to what we have masked, again using the image data. And in the second option, Imaris will calculate the cross-section area using, again, this mask, and then we'll try to approximate the diameter according to that. And of course, now the main question is, okay, so which algorithm is best to use in which option? And what I generally like to say is, or what we generally recommend is, if this path, what we have just created, is more or less nicely centered around the mask that we can see at the moment, I would say that the shortest distance from distance map would be correct. However, of course, optimization is always necessary, also always good and required in Imaris. So you can try both options and see which one works best with your data set. I will keep this option here, and I will click on next. And here we finally have our model. Now, what I want to do, and what I usually like doing before I continue on to detecting spines, is to see if the model looks good. So I would say that this looks really good for me. The only thing, however, that I can see here is this little area here in this little segment that has not been detected properly for some reason. So I can suppose, or I suppose what happened is that we didn't really have enough seed points here, and the seed point that was at this filament was rather closer to the filament right here rather than its true filament. However, there is a very easy way that we can fix that in Imaris. And what I need to do before I can do any editing, I need to finish, or I need to finish the wizard. So here I would press on the green button without looking at the spines yet. So I will finish the wizard, and this would be again the model. And what I can do now, so I have an array here of tabs that I can use, and there's different stuff that I can do with them, and we will go over more or less all of them in a second. But what I want to show right now is specifically this edit tab. And in this edit tab right now, by default, my mouse would select a segment. So that means that here, if I click on this segment, or if I click on this area, the whole segment will be selected. And I can just easily delete that, just clicking this on the selection delete option. And we have removed the segment. And that's the easy part. What's a bit complicated now, it would be to delete this bridging area without really deleting this continuation, because here the signal was segmented more or less right. The problem is that instead of its true branching point, it was kind of here wrongly segmented. But again, this is a very simple fix in Imaris. I just need to make sure that my mouse would select a point in my filament. And then what I'm doing here is I'm trying to get as close to the base as possible. And by holding down the control button, I can choose for another point. And now I have 2 points selected, and I can delete both of these points. And as you can see here, we have created this segment here that has been disjoined from the rest of the filament. And now, just like before, quite simple, I select for that segment and I delete it. The last step would be to connect this filament that we have created with its correct branching point. And again, in point mode, I will try to select the branch point that is more or less the true branch. And here again, by holding down the control button, I can choose the point that I want to then join this branch to. And here I'm finding the join button, press on that, and then Imaris immediately finds the proper filament. Okay. All right. So again, I will just fit and reset. Okay, so now that I am really happy with the segmentation and the modeling, what I can do is go back in the creation wizard. And for that, I need to click here on this magic wand. And once I click on it, I can already see all the creation parameters that we used to create these filaments and to calculate the diameter. Now I can go back at almost any step of the creation parameter. Let's say if I wanted to tweak something around, if I really was unhappy, I can go back and tweak those parameters. But in my case, I am happy and I just need to go and add some spines. So for that, I would click on the spines button. And just like before, here we have the option where we need to select the source channel, which again is the same one. And similarly, we need to select or to put in value for the seed points. So here it would be for the thinnest diameter or the smallest spine head. Now I can go back again and just kind of try and estimate a number. So this here I get 0.53. I would of course do this on a larger sample than just 1 or 2, but let's say I would estimate something about, so this number would be about correct. So 0.45, I measured around 0.5, so I will keep this value here. And the maximum length, so that would be the maximum length of the spine. And I think 11.4 microns is quite long, especially if we can imagine that 11.4 microns here, by looking at the scale bar, 11.4 microns would be really, really way too long for a filament or for a spine. So I think I will just use 3 microns here. And if you have spines that are branching, you can tick this option. In this case, I don't think that there are any branching points, so I will keep this option unchecked. And now I will click on the next button. Again, we have this histogram that we can increase and decrease the threshold for our seed points. And let me just make sure that we get those more or less correct. Again, I will try to zoom in, make sure that I'm getting the spine heads. Yes, more or less, I would say this would be good. I would click on the next button. And the next step would be to calculate the diameter of our spines. And we will have the choice between the same two algorithms that we already saw when we wanted to calculate the distance of, or the diameter of our filaments. Exactly, we can again choose between the shortest distance from distance map or the approximate circle of cross-section area. In this case, I would say that I would use the same short distance from distance map, and I can set up my threshold. Okay, and once I'm happy with the threshold, I will click on the finish button. Okay, great. So I will again reset my image, and I will switch off here the channel, and this is now the final model of this pyramidal neuron. By default, when it comes to the visualization style mode, we are using the cone mode, and that basically means that we can visualize the diameter that we have calculated again from the image data. However, if you're interested, or if you just want to visualize your filaments in cone or in cylinder style, you can also do that. Or if you want to strip down almost all information and just want to visualize it line style, you can also do that. But since we have calculated the diameter, I will keep on the cone version. We can choose to hide or show the spines. We can also, in this case, we can show the branching points. We can also show the terminal points like so. When it comes to these two, so rather more the edit tab and the draw tab, this is again a topic that will be more covered next week. So I will skip this topic. I will directly go into the filter. And here what we can do is we can filter using different parameters. So we can filter it based on dendritic area or branch level or whatever is interested. So for instance, we can only look at longer filaments, or we can only look at higher branching hierarchy filaments, and so on. So whatever we're really interested in. Here is the statistics tab, and I would say that this is really the most important tab for us. I would say right now, if I go to detailed mode, here again I can either get specific values or average values. Okay, and the reason why some of the rows here are highlighted in gray is because we have selected for some of the filaments here in the filter tab, and as we can see here, these highlighted filaments are now in yellow. And for the statistics here, they're actually highlighted in lighter gray. And since we were interested in, let's say, the branch depth, again, we can find all the information here. You can also find the dendritic length. If you're only interested in specific points or specific segments, you can then go ahead and choose them. Another very useful tool for visualization and comparison of the statistics here is, of course, if we do some color statistics coding. So if I click on the option here, so here we are in the color tab, what I can do, so for instance, in this case, I have chosen the object type to be the dendrite. Of course, I can choose the object type to be spines or branching points or spine branching points, whatever we're interested in, and we can then choose the statistics type. So by default, we started with the dendritic area, but we can, of course, use this color statistics for branch depth, or here as we were interested mostly in the length. And what we can see here, so I'll just move the display adjustment window a little bit. We have this nice color map that gives us some information, again, for the color coding. So the longer dendrites here would be in red, where the shorter ones here would be in this violet spectrum. Okay, so before I proceed to the next point here, which would be spine classification, I will just go back to base color. I'll actually get rid of this, and I will again just quickly here reset my image. Okay, so the last option here that I would like to talk about, this would be found here under the tools, and we have different extensions here that are listed, but I'm interested in classifying spines. So once I click on that option, Imaris and MATLAB would kind of bridge. Here we already have this MATLAB runtime compiler running for us, and what we can see here is that we can classify the spines in 4 different groups, or in 4 different types, where for each different type we already have some predefined parameters which we can modify if we like to. We can even modify the color, but in my case I would not modify anything, so I would click on classify spines. Okay, so here we can see again the progress. There we go. So I will just hide this window here. And here in different colors, we can already see the different types of filaments, or the different types of spines that we have classified. Now what I would like to do here is to just change them again. So right now we see them, or they're being visualized in this line mode. However, I would be interested again to visualize them more properly using the diameter that we have already calculated. Okay, so how about data sets that are not so beautifully and, so to speak, pristinely stained, like in this example? So what about those examples where we have some specks, or we have some background noises, or what if we're interested in segmenting more than one filamentous structure? So in order to show you that how we can do this in Imaris here, I will very quickly swap the example here. There we go. Okay. This example is of a mouse brain here, and this data set was provided to us by Dr. Matthew Coffron. And what we have here are 2 different structures, and in red we can see the vasculature, and in green what we have are the microglia. So a little bit of background information. The microglia are the central nervous system macrophage cells, and what they do is that they mediate the immune responses, specifically also in the brain. And their morphology is different according to their activation state. So if we have an unactivated microglia, they would appear more ramified, whereas the activated or reactive microglia would have much fewer projections. So for instance, this one here, so there are much fewer projections, and also larger cell body. And the reason why the non-activated microglia would be ramified is because they would extend their projection to kind of scout the milieu for different pathogens. So in this image, we will classify the microglial cells based on their morphology. So we'll definitely look at parameters such as branch length and number of branching points, number of terminal points, and also what we can look at would be the number of shell interactions. This way, we can determine the state of activation by quantifying the branch complexity and length. Since we have clearly starting points for our microglial cells, we can then easily use the automated autopath algorithm. However, in the case with the microvasculature, we would need to use the threshold algorithm, as there are no starting points and there's also a lot of looping structures that are continuous. Okay, so firstly, let's focus on our green channel or our microglial cells. So the first thing that I would need to do here is to create isosurfaces using the signal from the green channel. And then I can mask those surfaces and create a new channel here. So as you can see, I have already done that just for time considerations. So here I can show you what I have created previously. Let me just quickly switch this one off, and I will again center my image. I will not go over the process on how to create surfaces, because this has already been covered in the previous webinars. However, what I've done is I have filtered out some of the smaller surfaces just to make sure that I would get rid of any type of unspecific labeling or specs or background intensity information that I'm not really interested in. And in this case, I have also color-coded my surfaces according to the area where microglia with larger surfaces would appear lighter, and the microglia that have smaller surface area are in the lower end of the red spectrum here. Once I have created the surfaces, so here I can already click on this object. What I did was, by clicking on this button, Mask All, I chose the channel that I would like to select to create my new channel. So in this case, I used the channel 2 because that was the channel that was used for the microglial cells. And what I did was I simply set the voxels outside of the surface to 0, and then I pressed OK. So again, that I have already done, which you can see here. So let me just switch all the surfaces here. And now this is the cleaned channel, which then I will use to create my filaments. And just like in the previous example here, I would click on Add New Filaments icon, and I will again choose the autopath. So here I will go a bit faster because we have already went over the creation wizard once. What I need to do, however, is to make sure that the selected channel here would be my mask channel, so exactly what I just used. And here for starting points, again, we would be looking at the diameter of our soma, and the seed points again would be kind of the smallest diameter of our projections. And I can estimate these values here. I will just do it again quite quick. Rather, I would see here a distance of around 8.7, specifically for the cell somas, or around 9. I can also go deeper within my slices here, so I can take measurements from different Z stacks, but it looks like the distance value here is around 8.5. And when it comes to diameter here, let's see, I'll just zoom in. Let's say here it's around 2 microns. So go back to my 3D view, and here I will use the largest diameter. Here would be, as we said, around 8.5, and here the thinnest diameter 2 was the approximation that we got. And now I would click on next, and at this point it would take a little bit longer for Imaris to be able to find all the starting points. So as you can see, it's not really a problem for Imaris to find those starting points quite fast. And just like before, we can increase or decrease this value here, the number of the starting points. And also when it comes to the seed points, so here I don't really see as many, but I can here again use the threshold to then make sure that more or less I have selected all the seed points that will be then used by the algorithms to calculate the shortest path. Okay. I would say, to me, again, just looking at it by this already looks quite nice. Again, I have the same options, which I will skip again at the moment. And in this case, when I press now on the next button, the algorithm will commence, and it would generally, it doesn't take really too long. However, again, for time consideration reasons, I will directly show you how the result looks like. So this is where I have what I've done here. And again, I also used the calculate diameter option. Okay, so let me just highlight this filaments object. And I would say, let me just quickly again zoom in. I would say that the segmentation again looks really good. And what I would do now for visualization purposes, I will switch the visualization style to align, and I will switch off this channel. And here I will make sure that I can see the dendrites, I can see the starting points, but I also want to be able to see the branch points. So here you can see those in orange. And I would like to also use the terminal point option, which are then here nicely depicted by these green spheres. Now, hopefully you also noted that when I turned on and off this branching option, the majority of the branching points appear in yellow. However, we also have some branching points that appeared here in this navy blue color. And that simply means that at these branching points, there are simply more than two branches that are sprouting from this point. Okay, I will center again my image. Okay, so it would be now important to look at our statistics. And I would like to now go at the details tab. And instead of dendrite area here, I will look for a slightly different set of parameters. For instance, again, for the filament number of branching dendrites. I can also here find the statistical analysis for the shoal interactions. And when it comes to changing, let's say, the value for the shoal sphere's resolution, what you can do is, if you click what I just did, if you click on this wrench and screwdriver icon, what that does is that opens the statistics window in Imaris. So again, keep in mind that not every statistic is being calculated, not every statistic is being displayed. And feel free to, of course, tick or untick any of those statistical parameters that you would be interested, but you can also change the shoal sphere resolution here. Okay. And finally, to export your data, there are two different options where you can export the statistics that are displayed on the tab to file, or you can simply export all the statistics to a single file of your choice. How about now visualizing the vasculature? And just for a refresher here, so far we looked at these models for tree-like structures, so neuron and microglia, and right now we will switch gears a little bit and we will look into the second algorithm, which would be the threshold algorithm. Now what I will do here is switch off my filaments 1 objects, and I will turn on the channel with the vasculature signal. And similarly, like before, what I did is I already created some surfaces and I've already masked the channel. However, what I wanted to do before I would mask the channel is to make sure that I rather create more of a binarized channel. And the way to do that is again by clicking on the object that I would like to use for the masking. I would then click on the Edit tab, and then under Mask All, I would still keep the set voxels outside of surfaces to 0. But in this case, I would actually want to set my voxels inside a surface to some average value here of, let's say, 100. And as you can see, I've already done that, so I will not redo the calculations right now. But instead, what I will do is I will then show you how then this mask channel looks like. And this is the original data, and then this is again how the mask channel looks like. And really the reason why I'm doing that will become apparent in a minute. Okay, so I will take now the same steps that I've done previously. So I will start my filament creation wizard. And in this case, as we're having a looping structures, what we will do is we will use the threshold algorithms. Again, here's the description if we have forgotten what exactly or when exactly to use it. And then I will go on to the next step. Now, this is the important part. We need to really choose our source channel, and we have created this masked channel. So I will obviously now use this one. And here we have the option to enable feature preprocessing. And strictly speaking, this is not really necessary since we will be using this binarized channel, and this is really why we created it. And the enable feature preprocessing option is helpful when we want to detect pixels with lower intensity relative to higher intensity pixels. Furthermore, we have this option preserve edges. And this is another type of Gaussian filter here that will filter the edges around the dataset. But in this dataset, this is not really a necessary option. So let's presume that the original intensities in my original channel, the intensities in my original channels would form more or less a uniform signal. We also have a low signal to noise ratio, and there aren't really that many specks or unspecific staining. So in that case, simply going ahead and using this feature preprocessing option would be a good idea. However, in all other options, we would generally recommend to generate first this mask channel, what we just did previously, and then use that one. Now, just for demonstration purposes, I will have here the source channel as the original channel, and I would click here on to the next step. Okay, so here we have additionally an option to fill the cavities. So this is useful if somewhere in the middle there was a cavity, again due to staining, you could fill those so that you get a fully enclosed volume. And here we also have the option to use a connective baseline, where we can see in this intensity histogram we have two thresholds. So we have the lower threshold, which would be here this yellow line, and we have the upper threshold, which is the red line. And we could change those by dragging the line, so with the different mouse buttons. So for instance, for the red line, I would keep my finger on the right button, and for the yellow line, I would keep the left mouse button pressed on. And really the idea is that the voxels with intensities below the lower threshold will be considered as background, and the ones for the upper threshold, that would be the minimal intensity value of the object to be included in the filament calculation process. Or in other words, we will get a continuous looping filament structure when the gray areas were connected and also contain some of the white areas. So I would say the settings would be that I would use would be around. So in any case, making sure that all the signal then is being encapsulated by this gray line would be the most important step here. However, in our case, so I will just take a step back so we can easily do that. Again, in our step, we have, in our case, we have already created a mask channel. So I will then go ahead and use that one. Okay, and here you can already see the difference because we're using this binary channel. It is really not as important as to where we have set these threshold as long as we have detected all the original signal. Okay. All right, so I will proceed to the next step. Okay. So finally, in the step 4 of 5, which is what's called the graph completion, we can put in a value for the minimal ratio here of the branch length to trunk radius. And again, here I will use this show help option. And in this schematics, you can see the definition of the branch length and radius. And the idea is that if the base is rather thick and there are some short branches that are radiating from it, these will be deleted if we set a rather larger value, such as, let's say, 3 or 5. Okay, so in this case, let's say I would use a value of 5. And since there are no dendrite beginning points that I would like to detect, I would also switch this option off. Once I click here on the next step, the calculations, so the algorithm calculations will commence. And again, to save some time, I will not really perform it at the moment, but I will rather show you the finished results. However, the last step, as you can see, this is not really absolutely the final step of the wizard. I just want to let you know that the last step is where we can then delete this working channel that we have created. So once I start this calculation, what you would see, or what we would then be able to see, is that the result would look like this. And I will switch this channel, the working channel, off. And I will also switch here the mask channel. So this is how the final result would look like. Again, I can click on the filaments objects and choose the cone visualization mode, where here I'm already I have already ticked on the options to visualize also the branching points. Of course, this option for the beginning points will not result in any changes because there are no beginning points, but here we can definitely then see and visualize all the branching points. Again, we can then find all the statistical parameters that might be of interest under the statistics tab. And I can then go ahead and use my color tab to color code based on any statistics of my choice. So in this case, by default, we have the dendrite area, where we can visually appreciate the area by using colors of this spectrum color map. Okay, so these were really the 2 examples that I wanted to show you today. And with that, I would like to thank you for your attention and invite you to ask us any questions you might have in regards to the filament tracer. Thank you.
Okay, great job, Gergana. Thank you so much for going through Filament Tracer and how to use it with both neurons as well as other types of objects like vessels and that sort of thing. I think you've given a great introductory overview for everyone. So let's make sure I can still hear you as we jostled things around a little bit so that you could show your screen if you need to. Yeah, so yep. Okay. And we had a few questions come in, and we actually had a couple of people asking something pretty similar. So Orly and Mike were both wanting a fuller explanation of seed points. What exactly are they? and what are they doing, and what are they doing? And so could you go into that a little bit more and talk about seed points?
Yeah, sure. So I hope that everybody can see my screen. I should be sharing it at the moment.
Yep. Mm-hmm.
Okay, so I guess we are more here focused on the autopath algorithm, and essentially what the seed points are points that we add along the filament, and those are so basically the filament that we want to segment, and we add them based on the intensity information, and they are really necessary since the autopath algorithm will use them to create the shortest path. And if kind of we think about it in a different way, it's kind of like connecting the dots, where the seed points are the dots or the points that or through which the center line or the path would pass through. And I would say the most important aspect for the seed points would be to make sure that the ending points are well detected. So these would be the ending points here. So we need to make sure that these are well detected, because the line will follow the image intensity to create the shortest path at the end. So yeah, the ending would be the most important seed points.
Mm-hmm. Yep. Okay. I wrote all these down. Okay. And then we had one that's more about, and I think you were probably prepping for that. Sorry, I switched them up on you. I always say I'll do this in a good way and make it hard for everybody. So the next one is asking about when you're tracing something like vessels and you're interested in tortuosity or straightness, is that something that Imaris reports? And what are some ways of reporting or visualizing those changes in straightness throughout your image?
Yeah, sure. So yeah, as I also mentioned in my presentation, so that there are statistics which describe straightness. And here I can already show, so you can obviously find it in the statistics, but I can also show it here since we're under the color tab. I can already pretty much find it here. So when I go under dendrite straightness, I can see that the dendrites that are more straight, so in this case would appear here in the more reddish, where the ones that are not will be more towards the blue spectrum of colors.
Yep. And then you can also export that out as a table and work with it in Excel if you need to. But yeah, of course, seeing it as an image is always super helpful. So I just wanted to thank everyone again for attending the webinar, and to thank Gergana for presenting for us this week, and also to Rebecca and Dieter behind the scenes answering questions and taking care of everyone. Again, if you have any questions, try and get those into the question box. We'll leave it open for maybe another couple of minutes before we completely shut down the webinar. But I appreciate everyone coming. Thank you so much, and we will get back to you soon if you had a question. Take care and enjoy the rest of your day and the rest of your week. Bye, everyone.
Thank you. Bye-bye.